All right, today I want to go over some of those problems I gave you last time from chapter 10 that I mentioned 13, problems 13, 15, and 21. So I thought we'd walk through those. And so when chromosomes have got rearrangements like translocations or inversions, and they try to line up during meiosis, they're going to pair up in odd way to try to make the similar sequences synapse. So if you had chromosomes like the question showed here, you have a translocation. Part of this chromosome is over here, and part of this chromosome is now over here. So what type of translocation would that be? There's three choices. Reciprocal. And when they line up, they're going to matching sequences. So if we look at these are already pretty well lined up. I didn't put a dot, I put a dash for the centromere. So they're all lined up except when you get to here. So these chromosomes are going to try to line up right next to them, but the S and T are going to try to pair with the corresponding part of this chromosome. The IHGFDC, that all pairs up good, but the JKL and the JKL here are going to try to match up. So we could redraw this. I'll try. I'll put a dot for the centromere now. So they're going to form this sort of cross structure where now the T and the S are paired up, the J, K, and L are paired up, all of the M through R and all of the I through C. Everybody is matched up as well as it can with its the homologous genes, but the homologous genes are not on the right chromosome. So now the problem occurs when the chromosomes do segregation during meiosis 1. Usually homologous chromosomes would be separated. So that means that this chromosome here and this chromosome here, the MN, let's call them MN chromosomes, they should go into separate cells. So normal segregation is going to pull them this and this apart from each other. And it's going to pull this one and this one apart from each other. Okay. Now, when it does that and these two get pulled together, okay, so when it's both going up like that, that would be called adjacent type 1. Okay. Another possibility is this chromosome, this MN chromosome would get pulled to a cell. And this chromosome would get pulled to the same cell. Okay. So it's a little hard to imagine, but what we're kind of looking at is if we took those chromosomes and we were looking at them flat like this, right? Now let's turn them and look at them like this. So the MN where I drew the arrow is going to go up. And the IHGFEDC arrow chromosome over here is also going to go up. So I'm going to draw this arrow like this. So what I'm trying to draw here is that this chromosome that's over here is going to go up. The chromosome over here on the bottom is going to go up, and the other two are going to go down. So if you're looking at your equatorial plane like this, then up and down, right? And the way we have it here, where they're drawn, would be like going back and front. So it would be splitting this way. So it could split it on either of those two planes. There's almost a 50-50 chance that it will do that. If it does that, then that's called alternate, because this centromere is going to go into one daughter cell. This centromere is going to go down into the other daughter cell. This centromere is going to go up to the same cell that the first chromosome went into, and this one is going to go down. Okay? So if we number the chromosomes, if we call this 1, 2, and 3, 4, then with this type of segregation, which we call alternate, you're going to get 1 and 4 in the daughter cell, and you're going to get 3 and 2 in the other daughter cell. With adjacent 1, you're going to get 1 and 3 in a daughter cell, and you're going to get 4 and 2 in the other daughter cell. Okay? Then there's a third possibility. The third possibility is that these two chromosomes here both are separated into a daughter cell, and these two chromosomes are both separated into a daughter cell. And so that direction, that type of segregation is called adjacent to. And it does not separate homologous chromosome centromeres. So these two centromeres are homologous, right? They're from the same chromosome pair, original pair, MNOP. All of these genes are the same. So these centromeres are the same. And so this adjacent to is not separating homologous chromosomes, actually. Okay? So you would end up with 1 and 2 and 3 and 4 in the daughter cells. Okay? Now, of these three different ways of doing it, these two are going to occur about equal and about 45% of the time, and this one's going to occur about the other 10% of the time or so. Okay? Maybe a little more than 45% of the time. This one's pretty rare, but this one and this one both will cause problems. So the question said, look, what does alternate give you? Okay, so alternate is going to give us 1 and 4. So that means that the genotype would have N, MN, O, P, Q, R, S, T from this chromosome, a number 1, and it would have a number 4. So it would have L, K, J, I, H, G, F, oops, centromere F, E, D, C. So one of every gene has managed to get into those cells. So you have a chromosome 1 and a chromosome 4, you're fine. And the other one would have been, the other pair would be 3 and 2. So 3 would be T, S, I, H, G, F, E, D, C. That's chromosome 3. And 2, M, N, O, P, Q, R, J, K, L. So you have one of everything. Again, you're fine. Okay, so those are viable. This daughter cell is viable. This daughter cell is viable. We're all good. Okay. Now, adjacent 1, the other thing that's likely to happen is going to put 1 and 3 together. So So you're missing some stuff, huh? You're missing, you got 2 copies of S and T, and you're missing J, K, and L. So that's no good. And the other pair, you've got whoops, it's a J. You got this number 2 chromosome, and you've got this one. So in this one, you're missing S and T, and you have 2 copies of JKL. So that's not good. That's chromosome 4. So in both those 2 situations, that's non-viable. And then the last one, adjacent 2, is going to give you back 1 cell that has this and 1 cell that has that, okay? Because you took this chromosome right here, 1 and 2, 1, 2, and you took 3 and 4 and separated them. So here you're missing H, I, G, F, E, D, C. You're missing a bunch of genes, and you have here you're missing M, N, O, P, Q, R. So you're missing a bunch of genes, and you have 2 copies of some things. So you have problems both ways. So adjacent 2 is non-viable. Adjacent 1 is non-viable. The only thing that's viable is alternate segregation, where essentially every other chromosome is going to go into a daughter cell. All right, so whenever you're tasked with looking at a pair of chromosomes like that, probably the first thing you have to do is like this. You have to go ahead and try to draw them so that the genes line up. And take a chromosome and take the chromosome that has the next most similar genes and just write them down. And it does not matter which one you call 1, right? But once you call 1, 1, then go 1, 2, 3, 4 like that, okay? Instead of 1, 2, 3, 4, okay? If you go 1, 2, 3, 4, just the order that it wouldn't really matter, except then you'll go, but you found 1 and 2 in the same cell. And if you number it 1, 2, 3, 4, then 1 and 3 have to be in the same. So anyway, it just matters the numbering. The numbering doesn't really, it's arbitrary. Once you pick a numbering system, the thing is these 2 chromosomes should have the majority and their homologous centromeres over here. And the other pair should have their homologous centromeres over here. Okay, that's the only real rule. And then everything else will just have to line up. Okay. So it might take a couple of stabs, but once you do it a couple of times, then you'll see, oh, okay, I have to put those together. The next question had an inversion for 2 chromosomes. So an individual would have this genotype, and the inversion is between there. So it's a pretty long inversion. And I didn't do a very good job of drawing a loop. But it says try to draw a loop. So what you have to try to do is draw 4 chromosomes lined up together. So start off, and you're going to draw each chromatid. So we'll try to draw chromatid 1. I better start over here. So you're going to draw your chromatid, and then you've got to make a piece that's a loop, okay? So I looked at this and I said, okay, there are 4 genes that are the same order. So I'm going to need a pretty long stretch over here. And my centromere is located over here. So I'm going to draw a circle for that, a dot. And I've only got 1 gene that's the same on this end, right? So I've got A, whoops, this is supposed to be a lowercase a. I've got A, B, C over here, and then D, and then the part of the chromosome that goes on the loop or on the hump for this chromosome is the part where there's recombinate, or where there's an inversion. So I've got E and F and G and H and I and J. I still kind of messed it up. And then K is over here on this part. Okay, well, so the sister chromatid for this, we're going to call it number 2, and it's going to be just the same. So it's going to follow that same curve as best you can. And those genes are identical on both. I'm not going to try to draw them again. It gets too confusing. All right, now we have the inverted chromatids. So we'll number that one 3 and 4. Okay, so it actually, I'm drawing them too far apart. It actually helps if you draw chromosome 4 first. So here's chromosome 4. It's going to come in here, and then it's going to go loop-de-loop like this. Okay? And then you've got to draw chromosome 3. So this is actually 4. Now I've got to squeeze 3 in here. So 3 is going to come in, but it's going to go on the inside of the loop. And come out over there. I should have skipped more space. All right, so its centromeres are going to be in the same spot. Now, the other end of the chromosome is important to number. Okay, I'm going to put the 2 right there. So this is 1 prime, 2 prime, 3 prime, 4 prime. Okay, so number all the ends of both ends of all your chromosomes. And yeah, see, that looks like crap, right? It might look like crap. And it's not really that critical to get it neat. It's just critical that you can at least trace the line, okay? So it asked 2 things. It said, what happens? Part A was try to draw that. Part B was, what happens if there was a crossover between F and G, right? Oh, so the other thing is we probably want to go ahead and label our alleles. On the other chromosome was A, B, C, D, but then it was inverted. So you're just gonna have to skip along, and you're gonna go, okay, there's J, and then there was I, and a little H, and a little G, and a little F, and a little E, right? And then finally back down here is K, little k. So yeah, between D and J, we just arbitrarily stretch the distance at way out so it fits to the loop, all right? No big deal. We just pretend. Doesn't happen in real life. We just pretend. Between E and K, we had to stretch it out. Okay, don't worry about it. But it said the translocation happens somewhere in this vicinity on the chromosome, between the loci for F and G. Okay, and that's where you had your crossover. So that means which chromosomes are going to cross over? The 2 that are the closest together. So between F and G, that's where your crossover is going to happen. The 2 chromatids that are closest together. And if you track them, it's going to be chromatid number 2 and chromatid number 4. Okay, and if you draw it like this, and the crossover is in this loop, it's always going to be between chromosomes 2 and 4. Okay, so the resulting chromosomes, chromosome 1 or chromatid 1, will produce just a normal chromosome in the same order: A, B, C, centromere, D, E, F, G, H, I, J, K. You'll get to know your alphabet really good. That means it begins on end number 1, and it comes out on end 1 prime. If we track chromosome number 2, where is it going to come out? So start with 2. It's going to have A, B, C, D, A, B, C, centromere, D, and it's going to have E and F because nothing happened yet. So E and F, and then it crosses over and it continues to the right. So you're going to cross over and continue to the right. So when it crosses over, it goes over onto chromatid 4 and it picks up the little allele of G and H and I and J. And it's still coming around. This is number 4. It's still coming around. It gets a little D. It gets another centromere. It gets a little C. It gets a little B. It gets a little A. And it came out where? It came out on 4. So your translocation is always going to be between 2 and 4. Okay. Now we pick chromatid 3. Chromatid 3 just goes on this inside loop, doesn't touch anything. So it's going to be in its original order, A, B, centromere. Oh, for Pete's sakes. Centromere, D, and then it's just its regular thing, J, and I, and H, and G, and a little F, and a little E, and finally a little K, and you come out at 3 prime. My bad. 3 prime. You come out right here. So we have to create that other chromosome between all the ends. So you're going to need a 2 prime end and the 4 prime end. So you start over here at 2, follow it in. So what do you get to first? You're going to get to a big K. You're going to get to a big J. Because you're following this chromatid right here. It's a big J, it's a big I, it's a big H, it's a big G. And then it crosses over. And it keeps going in the same direction you were going originally. So you're going to keep going to the left. So it goes to a little F, a little E, follows the curve, comes around, comes back to a little K, comes out to 4 prime, and there is no centromere on that chromosome. It never had a centromere. So these 2 chromosomes here, the 2, 4 and the 2 prime, 4 prime, are the results of the recombinant. The meiotic recombination. And this one will get destroyed. It'll break somewhere between the 2 centromeres during meiosis. This one will probably be lost because there is no centromere. So this is called dicentric. This is acentric without a centromere. And both of those 2 chromosomes would not end up in viable gametes, meaning they won't get passed on to the offspring. So the only chromosomes that would be able to form gametes are the original inverted chromosomes, inverted to each other. The other part of the question said, what happens if the crossover happens between A and B? Well, that means it has to happen right here, right at the beginning of the chromosome. And if that happens, you're not going to actually you'll create recombinant chromosomes that are just fine, okay? So I didn't really do a very neat job here, but I'll try to fit them in down here. All right, so chromosome 1 is the same. Because nothing happened to it. It'll come out at 1 prime. Chromosome 2 is going to recombine, but now who is its closest neighbor? 3. So you're recombining with 3 now, chromatid 3, not chromatid 4. So now you're going to jump from 2 to 3. So you're going to have a capital A, then you're going to jump immediately over to chromosome 3. And you'll get lower cases, little b, little c, the centromere, little d, and just follow it right on around. J, I, H, G, F, E, K. And you're going to actually come out on 3 prime over here. And that chromosome's fine, just has 1 centromere. It's got all the genes. Then you actually have chromatid 3 to start with. And the only gene that you have there before you recombine is A. Then you're going to jump up to chromatid 2. So you're going to have big B, big C, centromere, big D, and you're going to follow chromatid 2 all the way around. So D, E, F, G, H, I, J, and a big K, and come out on chromatid 2 prime. And then chromatid 4, nothing would have happened to it at all, so it will be the original rearranged version. And come out back to 4 prime. So the only time it ever makes a problem is if you recombine anywhere in the interval that is the inversion interval. So if it had recombined between E and F, F and G, G and H, H and I and J. If it had recombined between any of those genes, you would have had big problems. Okay. All right, so then the last question was, how can you use deletion mapping to map the order of a gene, not just where one gene is, which is what we have talked a little bit about in class, but could you actually use it to map several genes? And the answer, of course, is yes. So here you're doing a little bit of that process of elimination again. So you have a list, they gave you a picture of deleted chromosomes. So the white space is missing, the black spaces are what's actual chromosome, or the black lines are actual chromosome, and up top is a unmodified chromosome. So the thing to go look for is, okay, you have situations where you want to look for the fewest missing genes or the fewest present genes. So 7 and 6 are actually good ones to start on. So chromosome 7 has only one deleted gene because it failed to complement with A. So A has to be somewhere in this area. We can pencil that in because that's the part of 7 that's missing, and A is the gene that's missing. So A is in the missing spot. 6 only has one gene. And so that means that that one gene must be in that area. So we can put an E there. So now we know where 2 genes are. So now I'll go to some of the other ones. So chromosome 1 is missing a gene B and D. So it could be that we have B and D like this, or it could be D and B like this. We can't tell anything about their order yet, but we know they're both in that region. So let's find another one that's missing just B or just D and see what happens. So this one, 4, is missing some of that. Let's look at 4. 4 is only missing D. I mean, it's missing other things, but we're just caring about those 2. So which end is missing? This end. And which gene is missing? D. So D must be here. So now we know the order is BD. Now look at another one that should be missing B. Number 5 should be missing B, and it's missing B, D, and E. So that makes sense. So all the genes here, E, B, D, that makes sense. Okay, so then we figured out 3 of them. So we can stop looking at some of these. So number 4 is also missing A. That makes sense. A was down here somewhere. So A should be gone from that one. It should be gone from that one. So number 2 is missing A and F. So F must be over here somewhere. Okay. Number 3 is missing A and G. So that means F is actually way over here. And G, is it here or here? We'll compare back to number 2. Was number 2 missing G? No. Was number 2 missing this region? Yes. So G is not here. G is in the part that's missing from 3 that isn't missing from 2. Now, number 4 is also missing F, so that makes sense. And it's also missing this region, and it's missing G, so that makes sense. C is missing, C. 6 is missing a bunch of stuff. It's missing A, okay. It's missing B, okay. It's missing D, okay. G, yep. F, yep. And what else is it missing? C. Are any other ones missing C? So C has to be out here on the end. That only one missing that piece is 6. Boom, we're done. We got them. E, B, D, G, A, F, C. There's your order. You can write it up here if you want. Okay, it's a process of elimination. Genetic Sudoku. All right, so today we want to start on a new topic, which is DNA and being the genetic material. And there's a lecture up already on this. So I'm going to hit some highlights. But DNA is, and RNA can also be the genetic material, but only for viruses. That was the answer on the question, on the quiz. Some individuals and a time frame for sort of the discoveries that went along with DNA being determined to be the genetic material. Way back, a physician, isolated DNA out of white blood cells. But the interesting part of the story is not that he isolated from white blood cells, but where the white blood cells came from. Pus, from bandages from wounded people in some war that was always happening in Europe. And he noticed that, of course, back then they would actually kind of just wash the bandages and then use them on the next person until they died, and then wash them again and maybe wash them again and then use them on the next person. And he was interested in what was in this pus, and so it's really mostly white blood cells, and white blood cells have a lot of DNA in them because they have nuclei, but our red blood cells don't have nuclei, right? So he started isolating stuff out of them, and he found this stringy material, and he called it nuclein, and it, of course, was DNA. So he was kind of the first person to really isolate some DNA from nuclei of cells, and it happened to be from white blood cells. And we do that now. We isolate DNA. I don't know if you guys did it in Life 1 or not. We'll probably do it in genetics lab, but we'll usually use cheek cells, just a few cells, and yeah, you can pull out some DNA and then do some analysis with it. So it's pretty easy to do. Then you fast forward quite a few years, and this guy, Frederick Griffin, I don't know if you say he really discovered it, but he noticed the process of transformation, which we talked about, cells taking up DNA. He saw that with some dead bacteria, and the DNA that was released from them was taken up by living bacteria, and genes that were encoded in that DNA from the dead bacteria were then expressed in the living bacteria, transforming their phenotype to become infectious, where previously they were not infectious. He couldn't really figure out what was going on with that, but he coined the term transformation. Great. So people started trying to figure out what was happening with that process. And finally, by 1940s, this fellow named Oswald Avery and a whole bunch of sort of postdocs and other researchers he had working for him figured out that they could destroy all sorts of molecules in that mixture of dead bacteria and still get transformation, except for when they destroyed DNA. So when DNA was destroyed, no transformation would take place. So that correlated with DNA being the genetic material. And after a few years, by 6 years' time, most folks in the scientific community believed that that was true, that DNA was the genetic material. But a few questions remained, like if it's the genetic material, it has to be passed on to other generations. So after World War II and the production of isotopes that were sort of developed along with the production of things like, uranium and plutonium, they figured out ways to enrich and get P32 and S35 and I-128 and all kinds of other radioisotopes that we could use as tracing tools in biological sciences. So you could take and have radioactive P32, and DNA has phosphate on it. They knew that. So you could get your DNA to be radioactively labeled with a radioactive phosphorus. Amino acids like cysteine and methionine have sulfur. So you could label proteins that have those amino acids using radioactive sulfur S35, and a number of other types of like molecules, carbon 13 and 12 and so on and so forth. So Alfred Hershey and Martha Chase were a couple of bacterial geneticists and bacteriophage geneticists. And so they came up with the idea of, let's radioactively label the components of bacteriophage by treating bacteria with the radioisotopes. So they had P32 labeled phage, S35 labeled phage, and then they looked to see that the P32, which would be the radioactive DNA primarily, did enter the bacteria. But the most important conclusion was that the next generation of bacteriophage that bacteriophage infects, right? And then it goes through the lytic cycle and out comes a whole bunch of new bacteriophage. Well, that same amount of radioactive DNA that they put in, pretty much all of it came back out in the next generation of radioactive phage, or a large proportion of it. So they concluded that not only was DNA the genetic material, but yes, in fact, we can demonstrate that it has gone from a parent generation of bacteriophage to a daughter generation of bacteriophage. And they actually tracked it for several generations. They just used that phage and reinfect some new cells and see that, yep, the new phage are radioactive. So they knew that there was a conservation, that the DNA that went in was replicated, but the old DNA remained and was actually being passed on. And around that same time, in 1953, and a few years earlier, Rosalind Franklin had taken several X-ray diffraction crystallography X-ray photographs of crystals of DNA to deduce the structure of DNA. And she was actually kind of going on the wrong road because she was a very good X-ray diffractionist, and her experience had always been in inorganic molecules, and you grow these crystals and you want to make sure that you have nothing else in there. Namely, the other thing that would be in there would be water molecules, water atoms incorporated in your crystal, and that will diffract X-rays and give you bad results. And so she made sure that her DNA had to be very, very dry, no water in it. And when she did it first with water in it, she got the famous double helix picture, but she knew those crystals had water in them. And so she kept working on completely dehydrated DNA, which gives a very different structure. And she would figure it out eventually, but it was a much harder structure to figure out. She was almost figuring it out when Watson and Crick saw the other data, and Francis Crick said, oh, that's a helix, and it has to be like this. And other people had already put in like what they knew what the chemicals were. They knew sort of ratios of the chemicals. So it was sort of a puzzle that fell together in the laps of Watson and Crick. And they made the leap to a model. So over the course of that 100 years, from 1850s to the 1950s, you basically went from, hey, there's this gooey stuff in the nucleuses of our cells, to at early 1900s, actually, that gooey stuff in the nucleus of our cells actually probably is important for genes because it's on the chromosomes, to, yeah, and we can take that DNA all by itself and give it to another organism, and it will give it traits, and it is passed on from parent to child, and it has this structure. And what's the coolness about the structure is it gives you a mechanism. It gives you a possible mechanism. You say, okay, it's 2 strands. One strand would always serve as a template for its partner strand. So that makes an easy template for replication. And it could be copied from being DNA into being RNA using that same idea. The DNA could serve as a template for the RNA. When you make new copies of it, it is inherited. One of the original strands is passed on. We know that from the Hershey-Chase experiment. So you make a copy. It's going to be a very faithful passing on of information from generation to generation, but yet you are making, the cell is making a copy. And so there is the inherent possibility of an accidental change, allowing for mutations to occur. See, there were 4 criteria that were all suitably answered, and DNA can provide all this. So obviously the actual nucleotide sequence of the DNA provides information. That was actually the last part of the picture to be figured out of how it worked, but that does provide the information, and it is transmitted from generation to generation. That was the Hershey-Chase experiment, as well as the information was from the Oswald-Avery experiment. The process of replication was implicit in the structure. The double-stranded structure implies a way to replicate it faithfully and to copy it into RNA for expression. The fact that it has to be replicated and that those are chemicals and there's going to be enzymes involved means there's room for error, which becomes variation. So DNA can meet all these criteria, right? So Frederick Griffith's experiments were done with bacteria, looking at a transformation from these rough bacteria into smooth bacteria. These don't cause any problem. The immune system destroys them readily. These, bad news, not able to be destroyed, and they can grow in the lung, get in the alveoli, and kill the animal, causing pneumonia, which is just fluid in the lung. So it had the name Streptococcus pneumonia. Okay, so he went through those experiments and determined that something from the dead bacteria was transforming the living bacteria. He called it transformation. Whatever it was, quote unquote, transforming principle, that's what Avery went after. Okay, he did things like, I'm going to say one of these biomolecules has got to be the thing because that's what's in there. So we can just destroy different things, take these things apart. Avery was a biochemist, and one of the classic ways to approach problems as a biochemist is through reductionism. Take it apart to its basic parts and then reconstruct the parts to get the whole working thing, right? In the industrial world, they call this reverse engineering, right? Take a rocket apart and see how it works and figure out if you can make it, or take a watch apart or an Apple iPhone and see if you can clone it, or Samsung. So you have this sort of thing going on with scientists too, with figuring out the biological things. And he figured out every time they destroyed RNA, or they destroyed just lipids or carbohydrates, or they destroyed the proteins, or they destroyed just the RNA, it still was able to transform. The only time that it wasn't able to transform was when you destroyed the DNA. So DNA seemed to be the magic bullet, if you will, that was going into those cells, changing their phenotype. And then Hershey and Chase used bacteriophage, and you recall that a bacteriophage is real simple with just DNA. The ones they used had DNA for their genetic material, and they inject that DNA into the bacteriophage. Here's a bacteria with a bunch of bacteriophage all over it. Electron microscope picture. with false colored. Phage aren't really blue. E. coli aren't really, well, they might be brown. I don't know, but they would all look gray when you took that picture. And so they could make the radioisotopes P32 or S35 and use those. P32 is primarily going to just go with DNA because DNA has the phosphate backbone. S35 is not ever going to be in DNA because there's no sulfur there. So the only thing it could radioactively label would be proteins, specifically the amino acids cysteine and methionine. So then they would have to grow the E. coli in those radioisotopes and then infect them with bacteriophage. The bacteriophage make new offspring, which are going to be radioactive. Then they take those radioactive bacteriophages and they infect fresh cells. So the only radioisotope came from the virus, right? And then they took the cells and they separated the dead viruses, what's called the ghost, from the bacteria. And you can do that with a centrifuge and a blender. Blend it and then put it in a centrifuge. The bacteria go to the bottom really quick, and the bacteriophage don't because they're so much smaller. So you can look and see, okay, are my bacteria radioactive? When the phage were P32 labeled, the bacteria were mostly. When the phage were S35 labeled, the bacteria were a little bit, but not nearly as much. S35 went, okay? So most of the S35 went away. Here, most of the P32 stayed. And then they let it go for a second generation. It infected. They took the bacteriophage that these bacteria, separated them from the dead ghosts, and then let them go ahead and be infected and make new phage. And the new phage were radioactive. So where did that radioactivity came from? They didn't add any extra during this whole time. So the only place it came from was the original DNA that got injected into the cell. So it had to come from the parent phage. And when they did the same thing with the S35, nothing was labeled. There was no radioactivity of any kind at all. So the only thing that got passed from generation to generation was P32. S35 never got passed on. By the way, if you're looking for a great Halloween costume, go as a phage ghost. Ask me about my other Halloween costume sometime. So now RNA can be the genetic material. We said that there were viruses with RNA in them. And along around the same time, and later in the 50s, some people isolated the RNA from tobacco mosaic virus, which infects a whole lot of plants, not just tobacco. And they purified the RNA, and then they just scratched it onto cells on the leaf. They didn't even make a virus out of it. They just rubbed the RNA onto the cells, and an infection occurred. More viruses were made. So that demonstrated that, yeah, RNA can really work, and you don't even need the viral protein. You just need the RNA. All right, so that brings us to, well, what does DNA look like? What's it made out of? What are all these components we've been talking about? And of course, this should be just a giant review snore session of Life 1. DNA and RNA are obviously nucleic acids. That's the category of biomolecule they are, macromolecules. The simplest building block are the nucleotides. And then those nucleotides are, in fact, themselves a composite of 3 biomolecules together, 3 separate molecules joined together. But those nucleotides are joined to form strands. And the strands for DNA will be able to base pair, generally forming a double helix, although there's examples of organisms like viruses that just have a single-stranded DNA. DNA will interact with proteins, forming chromatin in eukaryotic cells, other structures like supercoiling in bacterial cells. Can RNA base pair with DNA? It's a yes or no question, I guess I should say. So who says yes? Let's hear it. Aye. Shout out. Aye. Aye. Okay. Let's hear it. Can't. Shout nay. Okay. Now explain yourselves. Some aye person explain why can it base pair? What causes base pairing? What chemical bond is forming when a base pair forms? Yep. And RNA and DNA, are they made out of the same basic repeating units? Yes. What part of the repeating unit forms the hydrogen bonds? The nitrogen base. And so they all have those nitrogen bases, right? So now let's revisit our question. Who thinks RNA and DNA can form a bond between two strands, a strand of RNA and a strand of DNA? Aye. And the nays? Good. We convinced you, or at least you were scared to say anything. But the answer is yes, they can very easily, very readily. Any two nucleic acid strands, be it RNA and DNA, DNA and DNA, or RNA and RNA, any two strands can base pair. The only restriction or requirement is that the bases on the two strands be complementary. So an A goes with a T or U, a G goes with a C. So you have that ability to form double helices with RNA as well as DNA, and that's a concept that you had, that many people have a misconception that RNA has to be single-stranded and DNA has to be double-stranded. So that's why I did that little exercise. Okay, that's a simplified way of thinking about things that's not true in the real life, in the real world. So what are the basic components of all nucleotides, whether they be RNA or DNA? Everything has a phosphate or phosphate groups as part of their structure. If you're DNA or RNA, the defining difference between the nucleotides comes here, okay, is the sugar. The sugar molecule of the nucleotide is the difference between DNA and RNA, okay? Nothing else matters. So in DNA, you must have the sugar deoxyribose, and hence the name deoxyribonucleic acid. And in RNA, it is the sugar ribose, hence the name ribonucleic acid. What's the difference? So most sugars have a hydroxyl group on every carbon. And in this case, in DNA, there's one carbon which has no hydroxyl group, so it's called deoxy. That is actually on carbon number 2. This is carbon number 1, 1, 2. So it's 2-deoxyribose. Now, regardless of which sugar is present, that sugar may be attached to any number of nucleotides. There are 5 nucleotides that make up nucleic acids in everything we know about. There are other nucleotides, okay? So this is not the only nucleotides, but these 5 are the ones that you can find in DNA and RNA. 2 of them have a structure that has a double ring, a 6-member and a 5-member cojoined ring. Those are the purines, because the base chemical purine is what they are built around. Adenine, guanine. Adenine has only an amine. Guanine has an amine and a ketone sticking off from it, okay? So those are the purines. The short name, the big base. The other 3 bases are of the category pyrimidines. They have a single 6 or hexagonal ring structure, 6 carbon, well, it's not 6 carbon, 6 atom ring, thymine, uracil, and cytosine, okay? So yes, you need to know the structure of these things. And if you can draw them, you can recognize them. Okay, you say, oh, well, I'll just look at it and recognize it. No, you won't. But if you can draw it, you will. Okay, so I encourage you, if that's a strong enough word, to draw them for yourself until you can draw them by memory, and then take some drawings and erase the names, and then quiz yourself on do you recognize the structure. Or do it with a friend or your cat or whatever. So the cat won't get very many right. I'm just telling you. It's a good ego booster to beat the cat. So the big name, pyrimidine, goes with the littler, I think of them as the littler bases. Thymine and uracil are almost identical. Thymine has a methyl, uracil does not. Otherwise, they're the same, and they function the same, right? A can base pair with T, and A can base pair with U. Another common misconception is thymine is not present in RNA. Oh, yes, it is. And uracil is not present in DNA. And that's kind of correct. It gets there sometimes by accident. That's a problem. And the DNA has to be repaired. So it's not supposed to be in DNA because it can cause problems. But it can get there. Sometimes we do it on purpose, to cause problems, to make mutations. But the idea that T goes in RNA, or T goes in DNA and U goes in RNA is not something to focus in on. That's an arbitrary thing of nature, but it's not a meaningful thing of nature. Cytosine is in all of them. And cytosine has an amine group on it. All right, so you hook all those together. You take a base and the sugar. Chemically, they're synthesized with the base being added to the sugar first. So in our cells, they make the base and the sugar together create something called the nucleoside. And then the nucleoside will have a phosphate or 2 or 3, generally 3, added to it to create the nucleotide. So you'll hear the word nucleoside, you'll hear the word nucleotide. So nucleoside just has a sugar, S, side, sugar. Nucleotide has a phosphate, the T, okay? So that's kind of where they got those words from. When you name them, it's the name of the nucleoside that becomes the name. So adenine plus a ribose would become adenosine. If it was deoxyribose, it would be kept deoxyadenosine. So the deoxy out front tells you you had a deoxyribose as the sugar. So the purines end in ene, guanine, adenine, and their nucleoside ends in sine, adenosine, guanosine. The pyrimidines will end in dene. So cytidine, thymidine, uridine. There is no imidine, but there is uridine. All right, D in there, that means it's a pyrimidine. All these little things help you keep them, help you memorize them, okay? When you add one phosphate, it's monophosphate, so adenosine with one phosphate, or 2, di, or 3, tri, okay? So ATP, the energy currency of the cell in Life One lingo. But all these things, all of the nucleotides are built. So adenosine triphosphate, deoxyadenosine triphosphate, all of them are built. Okay, so here's the picture. So you've got a base, adenine in this case, onto a ribose. So this is adenosine altogether. The base always goes on carbon 1. When you add the phosphates, they go on carbon 5. And you add 1, you're mono, 2, di, 3, tri. And each phosphate has a name. Oh, they're not on there. The first one is alpha, second one is beta, and the third one is gamma, the third letter of the Greek alphabet. Alpha, beta, gamma. So they even have names. the numbers, right? Who's in organic? Who's inorganic now? Who's had organic already? Okay, so you remember you number these things, right? Every organic chemical name is 1,5 dash di-tri-pentyl. Blah, 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 blah. So the numbering stuff is kind of important though. So these guys number their thing. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 1 starts at this bottom. 1, 2, 3, 4, 5, 6, 7, 8, 9. These were numbered 1, 2, 3, 4, 5. So if you say a number, now you don't know, did I mean number 1 from the sugar or number 1 from the base? So they added a prime. We just love these little prime things. So that becomes 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So the sugar is the prime number and the base just has the regular number. And which thing is the most important for determining if it's DNA or RNA? The sugar or the base? The sugar. So it should be prime, right? So this has a hydroxyl here, so it's ribose. If there was no hydroxyl, it would be, what would the name of this molecule be? If that was gone, just a hydrogen instead. Deoxy, adenosine, triphosphate, right? If all 3 of them are there. So, right, here's all the names listed out for all of them. A lot of times we don't care. We just want to talk about all of them in one lump sum. So we use abbreviation. Deoxynucleoside monophosphate or diphosphate or triphosphate, DN, deoxynucleoside. Which one? It doesn't matter, whichever one, any of the 4, all of the 4. Nucleoside monophosphate, nucleoside triphosphate, so forth. So we use those abbreviations. If we want a specific one, then we would just abbreviate it like that. D, little d, everything else is capital. Okay. When we write out the sequence of DNA or the sequence of RNA, we don't put all that. We just put 1 letter representing the base because it's the base that is the information. Okay. So we would just write that as an A or a G or a C or a T. Okay. But you need to know it has other parts to it. So DNA is built and RNA are both built the same way by enzymes that will attach a nucleoside triphosphate or deoxynucleoside triphosphate, whichever the case may be. Here we're doing DNA because we got deoxys. It will attach this, and the attachment happens chemically by catalyzing the 3' hydroxyl to attach to the alpha phosphate between the alpha and the beta phosphates. So you're going to break this bond between the O and the P and reform a bond between the O and the P. And breaking that bond, it's a high energy bond. Okay, who remembers from Life 1 what kind of bond that is between a phosphate to a phosphate? This is an ester, yes. And it will create an ester, yes, but this bond is not an ester. It's not, the word ester is not in that name because it's not an ester. The new bond that will be made, this is phosphodiester. Yep, see, that's already labeled, so I wouldn't ask you that one. Who remembers? Who was ever told? Who remembers who heard the term acidic anhydride? Nobody. Great. So acidic anhydride is what that is. Write that down. An acidic anhydrous bond. Those suckers are explosive. Lots of energy. Okay? Lots of energy. This is what causes spontaneous human combustion. No, that's just a joke. Don't write that down. I mean, that spontaneous human combustion is not a joke. That causes it is a joke. It's no joking matter. But that is a lot of energy. Okay? Creates a phosphodiester bond, and the energy to drive that is produced by the cleavage of that bond. Okay? Just like ATP, ATP energy is used by breaking the bond right out here between the gamma and the beta. Right? This one just breaks a different one, but those two bonds have exactly the same amount of energy. This bond has, you have to put energy in to break it. Stable bond. This one's also stable. Okay. So that enzyme, if it's building DNA, you're adding individual units to a growing chain. A big long chain of anything is called a polymer. Polymer, many parts. So the enzyme that makes a polymer should be called what? Polymerase. And if it's making DNA, then it should be named DNA polymerase, right? Not Charles. Perfectly good name, but they name all enzymes based on what the enzyme does. So DNA polymerase makes a DNA polymer, or DNA, right? A strand of DNA. What does RNA polymerase make? An RNA polymer. And they both are going to be made by using another strand as a template, because that's how you know the order of the bases to insert. So A goes with T, G goes with C, except for when they don't. And that's when a mutation is going to happen, which we'll talk about in a few lectures down the road. But as far as you know right now, that has to be true. All right, so know your structures.